Hello, what I'm going to talk about today is how we're using a special type of microscopy to look inside tumors and watch in living color what's going on, and from these insights try to get new ideas into how we can treat cancer better. So this work really comes from the knowledge of pathologists. One of the first things that happens when you have a tumor, or suspected tumor, or a piece of the tumor or biopsy gets stained and sliced up and put under a microscope. A pathologist will then look at it, and one of the things they are seeing is, as you can also see here, the tumors are not just cancer cells. The cancer cells are shown here in blue, but in addition to the cancer cells, you can see that all these immune cells that are intermingled with the cancer cells. There are also other cells not shown here, and of course there are blood vessels that are bringing in nutrients to the tumors. So one of the things we wanted to try to see if we could achieve was to bring this dead tissue live and try to understand how does these interactions between the cancer cells and the other cells contribute to the progression of the tumors, and how can we try to affect them so we can prevent the tumor from further progressing. So to do this, we took advantage of some basic discoveries that had been done by a scientist that was not really working in the cancer research field. So this work started by discovery by a marine biologist that identified a gene in jellyfish that makes these jellyfish fluoresce green under UV light. And then another scientist figured that he could take this gene, transfer it into living animals such as this mouse that under normal light looks like a standard white laboratory mouse, but when you expose it to UV light you can see that actually some of the cells in this mouse are fluorescent green, and now you can watch them and follow what these cells are doing in the living animal. And then a third scientist find ways to change this gene so it could fluoresce not just green but also blue and red. And what we can do with this is now we can label different types of cells with different colors and follow how they are working. And together these scientists got the Nobel Prize because this has been such useful work in many different research fields. So what we did was taking this technology and get the blue fluorescent color into the cancer cells. We got the green color into the immune cells, the inflammatory cells specifically, and then we used a red color to label the blood vessels. And now you can see in real time here what's happening inside a living tumor in a mouse. You can see how the immune cells, the inflammatory cells, are coming through the vessels here. You can see rolling through the vessels, infiltrating into the tumor tissue, and how the cells are starting to interact. So what can you use this technology to? Well, one of the things we wanted to try to see is what happens when you give chemotherapeutic drugs. Why are they working in some areas and not for other kinds of tumors? Can we get some insights from watching in real time what's happening that we can use to get drugs to work better? So what we're doing here is we're injecting the chemotherapeutic drug doxorubicin used in breast cancer, and it's labeled in red so we can follow it. We've labeled the blood vessels in green, and you can see right after injection of the drug all the blood vessels are lighting up in orange. But as I start this movie, you can see that rapidly the drug gets out of the vessels, and each of these red dots is a cancer cell that's taking out the chemotherapeutic drug. And the brighter red these cells are, the more drug has accumulated in the cells, the more likely we are to kill the cells. So when we watch these kind of movies, one of the things that was really striking to us was that there was so much variability. Up here in this area, you can see a lot of cancer cells are positive. They have bright red. They're very bright red, and these are the cells we later can see are going to die. But then there's a region here where almost no cells are taking up any major amount of the chemotherapeutic drugs. And of course, these cells are not getting enough drug in to kill them. So that made us think about, could we find ways to get better penetration of drugs into the tumors? Is that one of the ways that we can get drugs to work better, drugs we already have? Another thing we discovered through these images was in a region like this one where you can see most of the cancer cells have been killed. We used here a special dye that labeled only the dead cells, and you can see all these red cells have been killed by chemotherapeutic drug. We didn't kill everything. You can see that in these mouse tumors, like in human tumors, one round of chemo is not enough. These blue cells, cancer cells, are still alive. But what we discovered when we watched this is you can see now this enormous infiltration of the green inflammatory cells that are coming into these regions of the dead tumor cells and are intermingling now with the cells. And when you watch a movie like that, and I remember it as yesterday, it was 4 a.m. in the morning when we saw this actually, the first question is, what does this mean? Is this a good thing or a bad thing? So to address this, we took a genetic approach and started looking at what happens if you take the inflammatory cells away from the mouse versus if you leave them in. So this is the normal response we get to chemotherapeutic drug in mice that have a normal inflammatory response. You can see here, even with chemotherapeutic treatment, the tumor continues to grow. It doesn't grow as fast as if we give it no treatment, but it still grows and progresses. And we have to ultimately euthanize the mice. But you can see here in mice where we have reduced the inflammatory cells, taken most of them away, we can now keep the tumor stable for a longer time, and these mice now survive longer because the tumor doesn't progress as fast. So what I want you to take away from this is that we now know that there's many different ways that we are not getting drugs to work as well as we could. There are ways where the cancer cells can acquire mutations and change other genetic changes so that they're not responding as well to drug as they should be. So there may be a single cancer cell in the middle of the tumor that's not really responding, and it's hard to see how you can do much about that. But in addition to that, we are finding that there are cancer cells that could have been killed. They don't have these mutations, but the drug is not getting in sufficient concentration to kill them. And interestingly, earlier this week, there was a group at Fred Hutchinson's Cancer Research Center that found a way to, in a subgroup of pancreatic cancer patients, modify the tumor tissue so they could get the drug to penetrate deeper into these pancreatic cancer tumors. And by doing so, they doubled actually the time that the pancreatic cancer patients survived after chemotherapeutic treatment. So that's something we in the field are very excited about. In addition, I showed you that these inflammatory cells are coming in and they're providing survival signals to neighboring cancer cells, and that too can make cancer cells that otherwise should die resistant to the drugs. So now there are similarly clinical trials ongoing to try to see different ways of inhibiting these inflammatory cells and see if that too can increase the survival and the response to a chemotherapeutic treatment. The results from these trials are not published yet, they're not known, they're still ongoing, but the results from the various different mouse models that's been done by many different groups actually have been very encouraging. So I want you to take away that there's in the field a lot of hope right now. We're finding a lot of different ways to target not just the cancer cells, but also the tumor tissue as a whole, so that we can prevent cancer from progressing, and even we're getting some insights on how the whole body is working with the tumor. And with that, I'd like to thank you for your attention. I'll be happy to take any questions. Thank you very much.